We're back with the routine lectures. Welcome back to my routine lectures. I'm Dr. Bobby, and New Age Cosmetic White at Tech Healthcare Consultancy welcomes all of you back. So we're back with the subject of general recombination molecular biology on Unit 13 of the CSIR-NET syllabus, which is equally effective and valuable for those sitting the NEET exams and all the Institute of Medical Sciences exams, and other similar exams too. Now, what we see here is a figure which shows the different resolutions of general recombination intermediate in mitotic and meiotic cells. As shown previously in figure 556, general recombination begins when a double-strand break is generated in one double helix, green, followed by DNA degradation. Excuse me. And the strand invasion into a homologous DNA duplex, red color. New DNA synthesis, orange, follows to generate the joint molecule shown. And depending on subsequent events, resolution of the joint molecule can lead either to a precise repair of the initial double-strand break left or a chromosome crossing over right. The experimental induction of double-strand breaks at specific DNA sites has allowed the outcome of general recombination to be quantified in both mitotic and meiotic cells, though more than 99% of these events fail to produce a crossover in mitotic cells, whereas crossover is often the outcome in meiotic cells. In either case, if the maternal chromosomes differ in DNA sequence in the region of new DNA synthesis shown here, the sequence of the green DNA duplex in the region of the new DNA synthesis is converted to that of the red duplex, a gene conversion. Right, we're back to subject matter proper. So with either outcome of general recombination, the DNA synthesis is involved converts some of the genetic information at the site of the double-stranded break to that of the homologous chromosome. If these regions represent different alleles of the same gene, the nucleotide sequence in the broken helix is converted to that of the unbroken helix, causing a gene conversion. The yeast Saccharomyces cerevisiae exploits the gene conversion that accompanies double-strand break repair to switch from one mating type to another, discussed in Chapter 7. In the sequence, in this case, a double-strand break intentionally induced by cleavage of a specific DNA sequence of the yeast mating type locus by an enzyme called HO endonuclease. After DNA degradation at the site of the break has removed the old sequence, the missing genetic information is restored by synapses of the broken ends with the mating type cassette, DNA sequence of the opposing mating type A or alpha, followed by local DNA synthesis in the manner previously indicated to reseed the broken region of the chromosome. In fact, it is through a detailed study of this precisely positioned form of double-strand break repair that the general mechanism of homologous end joining was revealed. Mismatch proofreading prevents promiscuous recombination between two poorly matched DNA sequences. Was discussed formally, a critical step in recombination occurs when two DNA strands of complementary sequence pair to form a heteroduplex join between two double helices. Experimental in vitro with purified rec protein show that pairing can occur efficiently even when the sequences of the two DNA strands do not match well. When, for example, only 4 out of every 5 nucleotides on average can form base pairs, if recombination proceeded from these mismatched sequences, it would create havoc in cells, especially in those that contain a series of closely related DNA sequences in their genomes. How do cells prevent crossing over between these sequences? Although the complete answer is not no, studies with bacteria and yeast demonstrate that components of the same mismatched proofreading system that removes replication errors. Example figure 523. Let's have a look at that. This is about replication and aspects of it. Binding of mismatch, proofreading proteins, DNA scanning detects nick in a DNA strand, strand removal, and DNA repair synthesis. Right, and that has the additional role of interrupting genetic recombination events between poorly matched DNA sequences. It has long been known, for example, that homologous genes in two closely related bacteria, E. coli and Salmonella typhimurium, generally will not recombine even though their nucleotide sequences are 80% identical. However, when the mismatch proofreading system is inactivated by mutation, there is a 100-fold increase in the frequency of such interspecies recombination events. It is thought that the mismatch proofreading system normally recognizes the mispaired bases in an initial strand exchange, and if there are a significant number of mismatches, the subsequent steps require to break and rejoin the two paired DNA helices are prevented. This mechanism protects the bacterial genome from the sequence changes that would otherwise be caused by recombination with the foreign DNA molecules that occasionally enter the cell. In the vertebrate cells, which contain many closely related DNA sequences, the same type of recombinational proofreading is thought to help prevent promiscuous recombination events that would otherwise scramble the genome. Seen in 568, the mechanism that prevents general recombination from destabilizing a genome that contains repeated sequences. Studies with bilateral bacterial and yeast cells suggest that components of the mismatch proofreading system diagrammed previously in 523 have the additional function as shown here. This is the picture here of the mismatch detected at abort sparing and preventing recombination. Right. And a summary: general recombination, also called homologous recombination, allows large sections of the DNA double helix to move from one chromosome to another. And it is responsible for the crossing over of chromosomes that occurs during meiosis in fungi, animals, and plants. General recombination is essential for the maintenance of chromosomes in all cells, and it usually begins with a double strand break that is processed to expose a single strand of DNA end. Synapses between the single strand and a homologous region of DNA double helix is catalyzed by the bacterial REC-A protein and its eukaryotic homologs, and it often leads to the formation of a four-stranded DNA structure known as Holiday junction. Depending upon the strand cuts made to resolve this junction into two separate double helices, the products can be either a precisely repaired double strand break or two chromosomes that have crossed here. Because general recombination relies on extensive base pairing interactions between the strands of the two DNA double helices that recombine, it occurs only between homologous DNA molecules. Gene conversion, the non-reciprocal transfer of genetic information from one chromosome to another, results from the mechanisms of general recombination, which involves a limited amount of associated DNA synthesis. So that's it about this lecture portion here. Right, going on to the second portion of histochemical techniques. We'd like to first give all credits and express our gratitude to William H. Outlaw Jr., Stephanie Springer, Mitchell C. Tashinsky, Biology Unit First, Florida State University, Tallahassee, Florida 32306, for this invaluable abstract for reference. So going ahead with histochemical techniques. Biochemists who study single cells have been constrained by the lack of general methodology of high-term resolution with high measurement sensitivity for quantitatively assaying enzyme activities using natural substrates in solution. Once this is over, once I finish narrating this, we'll have the latest updates as well, so you know where you stand, right? And the methods we describe will remove this limitation. In brief, nanogram tissue samples are dissected from frozen dry tissue. The samples are extracted in microdroplets of assay cocktail. The enzyme activity, indicated fluorometrically by the oxidation reduction of NADP, is followed in real time on a computer display. In the development of this method, we evaluated several parameters required for optimization. The most important of these evaluations, including numerous empirically and derived relationships, are reported here in supplemental material provided with the reprints. Well, with these methods, assays of pyruvate orthophosphate dikinase on samples were enriched in bundle sheet cells and mesophyll cells of Flavariae brownii, yielded the predictable results. Assays of this enzyme in GOD cells dissected from Vicia faba leaflets gave results like those recently reported by another laboratory for protoplasts derived from the cells. The results of assays by these method and by enzymatic cycling for NADP triose dehydrogenase were comparable. Phosphoenol pyruvate decarboxylase and the most extensively studied enzyme activity was present in high levels in GOD cells, which has been demonstrated previously in other reports based on diverse assay approaches. That's about the abstract of this article. To the main article. In some instances, the activity of relatively few cells that are dispersed throughout an organ determine the functional competence of the entire organism. For example, GOD cells comprise about 0.1% of the volume of a leaf. Pairs of these cells surround stomata and regulate the aperture sizes. Yet the size of these pores is a primary determinant of the rate of carbon dioxide uptake by the leaf. To gain a biological understanding of the function of specialized cells, an investigator is challenged to develop or adapt appropriate methodology. Sometimes the cells can be isolated in bulk from the surrounding tissue matrix and studied by conventional biochemical methodology. This approach is attractive, number 36, but is limited to tissues amenable to maceration and unfortunately is potentially subject to artifacts arising from instability of cellular constituents. Alternatively, such studies may be approached using histochemical methods and in broad terms, these methods fall into two categories. A, those that rely on high morphological resolution of the assay indication, example electron microscope, slide histochemistry, and B, those that rely on sensitive analytical techniques for analysis of extracts of NG tissue samples that have been dissected from the surrounding tissue, example the Lowry methods. These existing techniques are valuable and reliable, and indeed have used many of them in attempts to characterize GOD cells biochemically. In the more specific case of quantitative assays of minute amounts of enzymes, investigators have been restricted to slide histochemistry, example stains, isotopes, and microdroplet chemistry, example the cycling technique, spectrophotometry, luminometry, fluorometry. Slide histochemistry is subject to artifacts arising from diffusion limitations throughout the tissue slide. Perhaps more important, interpretation of the results is confounded when non-physiological substrates are used, as in page 35. The use of microdroplet techniques allows one to avoid these problems, but typically assays are indirect. A product accumulated during a long incubation is measured later, or they attack sufficient sensitivity for high-time resolution. Thus, a method suitable for single cells and analogous to a conventional NADP-linked spectrophotometric assay, that is, automated, real-time, direct, quantitative, and using physiological substrates in solutions, has not been available. For this reason, there have been few attempts to characterize enzyme activities kinetically in single cells. and almost all cells validations of assay parameters for particular cells have been conducted using whole organ extracts in ml volumes. Right, we have here a direct method for enzyme assays using natural substrate in solution with a time resolution of seconds and the measurement activity of f-mole. The approach is based on computerized data acquisitions and analysis of photometer signals resulting from NADPH, fluorescence, and microdroplets. The concepts themselves are not novel. It is the application that should make them amenable to experimentation. Previously, practical biological studies techniques for tissue sampling were developed by Laurie and co-workers. Fluorescence and microdroplets has been reported by Rotman and subsequently by several others. In those studies, fluorescence was developed to determine, after a prolonged incubation period, real-time measurement of NADPH at a sensitivity of about 0.5 p-moles have been reported by Jonquin et al. The recommended method is a compromise between optimization, exam avoidance, photobleaching, sample high measurement. The general description of methods. The nanogram tissue samples are dissected from freeze-dried tissue and weighed on a fishbowl balance as described elsewhere. An assay cocktail, 1 to 200 nanoliters, is pipetted using a special constriction pipette into oil, which serves to prevent evaporation. Then an individual cell sample is pushed through the oil using a tiny quartz fiber with a gas handle. All right. Continuing on the general description of methods, the nanogram tissue sample is dissected from freeze-dried tissue and weighed on a fishbowl balance. And then an individual cell sample is pushed through the oil using a tiny quartz fiber with a glass handle until contact with the droplet is made. The tissue is wetted but remains at the cocktail oil interface. At predetermined intervals, the observation lamp shutter closes and the excitation lamp shutter opens. The excitation light illumination passes through a heat shield, the field diaphragm, condenser, and a wideband filter. It is reflected by a dichroic mirror to the objector and passes through the bottom of the sample chamber and into the center of the reagent droplet. Emission light is collected by the objector, passes through the dichroic mirror, a wideband filter, an adjustable measuring diaphragm, and an interface wedge, and is measured by a photo multiplier tube. The analog signal is processed by an electronic console, converted to digital and transferred via an input-output processor to a microcomputer. The signal is evaluated by a computer and the time of the reaction is plotted in real time on the display. At the end of the experiment, a hard copy of the graphics and data produced on the system printer. Other software can be used for consolidation of experiments and plotting. Materials and methods. Photometer equipment. The following components were supplied by Leeds: diode microscope stand, stage, binocular observation tube, eyepieces, lamp housing, transformer, diffusion disc, lamp housing, power supply, lamp holder cables, condenser, filter cube A, MPV compact, electronic console, iris diaphragm adapter, objective interference control unit holder, and adapter. The heat shield filter made for slide projection was from Kodak. Computer equipment. Following components were supplied by Hewlett-Packard: computer, RAM, mass storage, printer, plotter, and the rest of it other materials. The fava were cultured as previously described, 14 frozen. 80 leaves of Flavana brownie were kindly provided by Dr. Scott Holliday. Sources of other plants were kindly, as indicated in the text, before quenching in N2. Nitrogen leaflets were pre-illuminated. Then leaf fragments were frozen, dried, and samples were obtained and weighed as described by Lowry and Pazinot. In a conditioned room, 40% RH, 80 degrees centigrade. Analytical enzymes were from Boehringer, both. Most of the chemicals were from Sigma. Sources from Millennium suppliers are indicated in the text. So it's part of the research that's being done in this lab study. Phosphoenol pyruvate carboxylase, the acidic oxide was 6 to 10 nanoliters of 50 mm tris, pH 8.7 containing 0.1 mmEDTA, and all these substances. And we had NADP, triphosphate dehydrogenase, pyruvate orthophosphate dikinase. All our assays and our cocktails we use. Method evaluation: sample chamber design. Evaluation and emission light pass through the bottom of the chamber, and therefore its construction is critical. We have evaluated numerous film types which were received as a result of greater than 50 solicitations to US manufacturers. And although many films have acceptable optical qualities, most like other chemical resistance required for cleaning, thinness required because of the short focal length of lens of high magnification or an acceptable degree of hydrophobicity. They also use 3 sample chamber. Before that we have transmittance is somewhat higher for Teflon, DuPont, technical bulletin, and excitation light. It was a 325, 340, or 366 nanometer. And Bayer atomic ratio recording spectrofluorimeter SFR 100 was used. They also used the sample chamber configurations: a flat Teflon tray, 6 and 2 versions of modified oil well rack. The easiest chamber to construct is the tray, but reagent loss through evaporation when some droplets tend to position at the oil interface is more common than with it, and it is inconvenient to use. First version of the modified oil well rack is constructed as described by Lowry and Pazinot, except the wells are drilled completely through the Teflon blank. For this version, chlor, trifluoroethylene, Kel-F, native plastic, etc. is also acceptable and is easier to machine. Then the Teflon film is bonded to the entire lower surface using chem grip. Third, we emphasize second, as the thin film is not sufficiently heat resistant to permit over drying after cleaning. Third, it was emphasized that it's important that the film be taut when bonded, and it is imperative that the well bottom not be contaminated with bonding agent. The second version of the modified oil rack is the most difficult to fabricate, but is most satisfactory. In brief, wells are drilled completely through a Teflon block, generally as described in the preceding paragraph. Then a 1 by 32 inch at the bottom of the block is removed with a modified end mill, except for a 5 by 32 inch OD rim around each cell. The holes in the snap-on mylar sheet must be machined to very close tolerance to prevent all seepage from the wells. Light filtration: edgy lines group at 365 nanometers passed by the excitation filter, reflected by the dichromic macro mirror less than 400 nanometers. Filtration of fluorescence beyond that provided by the emission filter, long past 430 nm on filter cube is necessary. It's also recommended that additional filtration with adjustable interference wedge, half bandwidth at 25 nanometers set at 490 nanometers. Field diaphragm setting. The effect of the field diaphragm setting 5 settings over a range of 10 to 570 millimeter squared on signal by noise ratio and signal intensity was studied. Photo bleaching: NADP H is susceptible to photo bleaching. There are virtually infinite combinations of ways to reduce photo bleaching to insignificance for each volume example attenuation of excitation light neutral density, filters and choices of lenses, field, diaphragm settings, length of time the excitation light starter is open, frequently of readings. Then is about the diffusion with the recommended method. Fluorescence measures are made from the center of the droplet only, thus the time course of a reaction can be followed accurately. The demonstration experiments in the measurement precision sensitivity, NADP trials, dehydrogenase, pyruvate orthophosphate dikinase during stomatal meiopen opening, guard cells accumulate organic anions, starch concentration decreases. And the rest of it is all figuratives, which need detailed discussion if you're interested. And the conclusion is, as demonstrated in this report, direct assays of enzyme activities in a single cell can be conducted with a time resolution of seconds and a reading precision of f-mole. These techniques are an attractive alternative for current methods for localizing enzyme activities to the cell level. And more importantly, they may be used to conduct detailed kinetic analysis of enzyme activities in a single cell using natural substrates in solution, which has not been possible using other current approaches. It's also noted that the minute quantities of metabolites may be enzymically estimated using the direct methods, but this approach for metabolites is not an improvement over current methods in opinion. We have references and the literatures are cited. Please make references and make your notes. That's it for this lecture. Thank you.